Amphibians across the globe are experiencing a pandemic, a story that we can all relate to. However, while our pandemic is caused by a viral pathogen, amphibians are at risk of a fungal pathogen known as dendrobatidis, otherwise known as the amphibian chytrid disease. The chytrid fungus was initially discovered in the 1960s throughout North America, but studies at the time suggested that the fungus was not likely a new pathogen, as other studies state that amphibians infected with chytrid disease have been found as far back as 1938. Since its discovery, the fungal pathogen has been found throughout the globe, affecting over 500 amphibian species across 52 countries, causing a mass decline in amphibian species, which is being dubbed the sixth mass extinction. Chytrid infection varies throughout both inter- and intra-specific populations, which is due to variety within the host resistances, the type of chytrid strain infecting the host, as well as the environmental conditions. The fungus consists of two life stages, the first being an aquatic flagellated zoospore, which is an infectious stage. The second is a spherical thallus existing within the extracellular space of the host. This is the reproductive state. Chytrid affects the skin of infected hosts, preventing their ability to breathe, ultimately causing death. Throughout Europe, chytrid distribution is rather ununiform, but the UK had its first documented case back in 2004, though many believe that the pathogen first arrived in the UK sometime around 1998. In 2008, a survey conducted on UK amphibians across England, Scotland, Wales, and the Channel Isle Jersey confirmed that amphibians had tested positive for chytrid in all study sites except for Jersey. Another survey conducted in 2011 also confirmed the presence of chytrid disease throughout amphibians across the UK. To test for chytrid disease in Wales, two sample sites were studied, the first being Morphydifferent and the second being Treborth Botanical Gardens, owned by the Bangor University. In order to collect specimens for study, nets were used to dredge through the bottom of water channels and ponds and catch specimens. Once specimens were caught, they were tied off in plastic bags. This is important as it prevented specimens from coming in physical contact, as if they had, we ran the risk of infected individuals infecting healthy individuals with chytrid disease. After sample specimens had been caught, they then had their bodies swabbed. The entire underside of the animal had been swabbed. This included their belly, foot webbings, underside of the limbs, and the underside of the tails in salamanders and newts. Swabs collected epidermal cells and mucus samples from the skin, and in total 46 specimens have been sampled. From Morphydifferent, 12 toads, 3 smooth newts, 8 palmate newts, and 1 alpine newt have been sampled, and 22 palmate newts have been sampled from Treborth. To test for chytrid presence in sampled specimens, DNA was extracted from the used swabs for use in a qPCR, a process which is able to synthesize a full chain of DNA from smaller fragments of the original strand of DNA. This process is so sensitive that it is able to detect a single zoospore or thallus of chytrid from a given sample. To do so, cotton tips were cut and placed into microtubes. 50 microliters of a chemical known as Prepman Ultra was added to the swab tip. The tubes were then placed into a vortex, then a centrifuge, and then a vortex and centrifuge again, before being added to a heat block for 10 minutes. Once this 10 minutes was up, the samples were then removed and centrifuged again. The cotton tip was then removed, with the remaining liquid in the microtube being extracted DNA. A diluted DNA sample was mixed for each specimen. This was created mixing 90 microliters of ultra-pure water and 10 microliters of DNA extract. This was then again vorticized and placed on ice. In order for the qPCR to be successful, a series of chemicals referred to as reagents must be mixed with the DNA extract. The mixture was then placed into a PCR machine where the qPCR process took place. From this investigation, it was found that no specimen tested positive for chytrid disease at either of the two sites. While this is the desired result, it does not mean that the sites are in the clear, as now actions need to be taken to prevent chytrid disease from infecting either of the two sites. Measures need to be taken to prevent chytrid infection. One such way would be to have visitors to either of the sites disinfect their footwear, killing any chytrid zoospores they are carrying, preventing them from finding a host to infect. Restriction of pet trades will also help to prevent the spread of the disease, as non-native amphibians act as vectors of chytrid, so their reduction in homes will prevent infected pets from passing the disease into wild species through their owners, as well as prevent infected pets being abandoned in the wild, causing a further spread of the disease. Sadly, little is known about the best way to manage chytrid-free sites and to reduce mortality of individuals within infected areas. There have been attempts to immunize amphibians against chytrid disease, though these have been unsuccessful. However, it is possible to disinfect infected amphibians through heat exposure.